This video will focus on gene isolation and manipulation. Some of the concepts we'll cover are listed on the screen there. We'll first of all discuss how we can create recombinant DNA. We'll discuss how we can create DNA libraries, which are just collections of specific pieces of DNA. We'll discuss PCR, or polymerase chain reaction, and also mutagenesis, a way by which we can create changes in the bases in DNA that can result in changes in amino acids in proteins. First of all, let me just mention that recombinant DNA has been around for quite a while. In the 1970s, people started having the ability to move genes from one species to another. And this has not been without controversy. Certainly over the years, questions have cropped up regarding the safety of genetically modified foods for human consumption, and also, of course, the safety of organisms in the environment and the impact that GMOs might have on other species out in the environment. And so many of the techniques that we'll discuss in this chapter, again, are used to create recombinant DNA. And if we talk about transgenics, we're just talking about basically introducing a gene from one species into another for various specific reasons. Let's first of all discuss about recombinant DNA in a general sense. And I have up here some comments about bacteria, because bacteria are used quite often in recombinant DNA technologies. So first of all, I just want to point out with bacteria, yes, they have a chromosome, but it tends to be a large single chromosome, and it's circular. And you see the main chromosome toward the left here, toward the bottom. But bacteria often also have these much smaller pieces of DNA called plasmids. And so to the right, we see a circular plasmid. It can contain various components. It might often contain genes that code for proteins that confer resistance to antibiotics. It might have an origin of replication, and that's important because when have plasmids replicate every time that a cell divides. And we also might have some cloning sites. These are regions where the plasmid can be split apart with various enzymes. And if we're interested in using this plasmid as, say, a vector to move DNA from one species to another, we can take advantage of these cloning sites and insert specific genes into those regions. So let's talk again in a broad sense about some of the techniques that we use for recombinant DNA technologies. So how do we create engineered DNA or recombinant DNA? Well, first of all, we isolate whatever it is whatever DNA we want to work with. That could be the entire genome, for example, of an organism. It just depends on the question at hand. So then we could take the entire genomic DNA from a species or an organism. We could cut it up with various enzymes, for example, or we can physically shear it by forcing it through a thin syringe. And then we essentially take all those pieces and then add them to plasmids. And that's what we see toward the right here. So a bunch of different plasmids, each with a different piece of DNA. And I just want to mention that a plasmid that is commonly used to create genetically modified crops is Agrobacterium. Agrobacterium is a bacterium that can infect plants and essentially can insert its genes from its plasmid into the chromosomes of plants. And when expressed, those genes code for growth factors that can cause uncontrolled cell division, like we see on the tree toward the left here. But again, we could take advantage of the plasmid in the Agrobacterium to introduce specific genes into crop plants, for example. And so it's called, I just want to mention that the plasmid is called the TI plasmid for tumor-inducing plasmid. Once we have a plasmid that has been engineered with, let's say, BT, Bacillus thuringiensis toxin, or let's say Roundup Ready proteins that can be introduced into crops like sugar beets, we can take the plasmid, add it to a cell culture of our crop plant in question. The plasmid will incorporate the genes into the chromosomes of the plant. We can take those cells and grow them up into a new plant, and we end up with a genetically modified plant. So again, Agrobacterium contains a plasmid that's often used to produce genetically modified plants. Just some key terms that I just want to mention as we go through this chapter, because these might come up occasionally. So we're talking about a clone. Clone, of course, just refers to a genetic copy of something. And so we can talk about cloning an entire individual, or in this case, we're talking about cloning genes, so making many copies of specific genes. Vector simply refers to something that can carry a piece of DNA. Typically, we're talking about carrying genes from one species to another. And we can use various vectors, for example, plasmids. That's what we're going to focus on throughout this chapter. If we talk about an insert, that just refers to a specific piece of DNA of interest. So it could be just a fragment of DNA. It could be the exact coding sequence. So for example, an exon. It just depends, again, on the procedure at hand. And so since plasmids are often used as vectors, let's just talk about some features of plasmids that make them good candidates for vectors. In other words, for moving genes from one species to another. First of all, we can have plasmids have multiple cloning sites. That's what we see. All these interesting little abbreviations toward the top here. Those are just specific sequences that can be cut with restriction enzymes, and we'll talk about those in a second. But again, these are regions that can be cut with enzymes to essentially break apart the plasmid in this region right here, so that we can then insert a gene of interest. Plasmids often have selectable markers. So for example, this region right here codes for ampicillin resistance. We'll talk about that in just a bit. And also, again, they should have an origin of replication so that the plasmid will be replicated along with the main chromosome within bacterial cells. So again, those are just some general features of plasmids that are used as vectors. Restriction enzymes or restriction endonucleases. So these are enzymes that cut DNA at specific sequences, and they recognize specific sites within DNA. And then essentially what we can use these for is to cut DNA apart, like we see here. We can then insert a gene of interest between these two regions. Again, that's essentially one way to produce recombinant DNA. So if we think about restriction endonucleases, we can end up with a variety of outcomes. Some enzymes give us sticky ends. That's what we see toward the first two here toward the top. So on one hand, we can have sticky ends with a 5' overhang, like we see toward the top right. We can end up with sticky ends with a 3' overhang, like we see toward the middle. And some restriction enzymes actually produce blunt ends, where we just simply see a cut straight down the middle between adjacent base pairs. Now let's briefly talk about DNA libraries. These are simply collections of many pieces of DNA. I illustrated here two different types of libraries. So toward the left, a genomic DNA library. This is essentially a collection of all the entire genome of a species. And it can be found as many small pieces of DNA, and we'll come back to that in a second. On the other hand, we might be just interested in the coding regions of DNA. So toward the right, we see a cDNA library. Again, this would essentially consist of all of the coding regions, so the exons only, of a particular species. So if we go with a genomic DNA library, you're going to have all of the components of genes. You might have promoters, enhancers, silencers, and you'll have introns as well as exons. But if you focus on creating a cDNA library, again, that's just going to focus on the exons, because as we're going to see, it's based on the mRNAs found within cells. So let's begin our discussion by walking through the basic events involved in creating a genomic DNA library. What you can do then is take the DNA from your organism in question, cut it using restriction enzymes, for example, add those pieces to plasmids like we see toward the right, and so now we have a bunch of plasmids, each of which carries a different piece or a different section of DNA. Then we can take those plasmids, use them to transform bacteria. That's what we see toward the lower right here. So now we have a bunch of bacterial cells, each containing a different piece of DNA. We can then grow those bacterial cells on plates, like we see here, and then theoretically you can end up with colonies, each of which can be formed from a single bacterium containing a specific piece of DNA from our DNA that was cut up to begin with. And so just imagine a situation, you've got agar plates with all these bacterial colonies, and so you have to ask some basic questions. First of all, how do you know which of those bacteria actually incorporated a plasmid? Because not all of them will. And beyond that, how do you know which of those contain a plasmid that contains a piece of DNA and not just an intact plasmid to begin with? So we can accomplish some basic selection procedures as follows. So for example, if we're using a plasmid, shown here in yellow, that contains a gene for ampicillin resistance or some other antibiotic resistance, if we then grow the bacteria on medium containing the antibiotic, for example, ampicillin, you can imagine, as we see toward the far right, any bacterial cells that did not take up a plasmid, they simply won't grow. So you don't even have to worry about them. On the other hand, the other two bacterial cells that we see here toward the left and the middle, they both have taken up a plasmid. So they'll both grow in the presence of this antibiotic. But how do we know which of these cells contains a gene that has been inserted into the plasmid? Toward the middle, we just see an intact plasmid. Again, that'll grow just fine. Toward the left, we see a plasmid as well, but notice this one actually has our gene inserted, or a piece of DNA inserted. So again, how can we distinguish between these two? Because obviously what we're interested in are the bacterial cells toward the left. Well, now we can take advantage of another marker. And one that's commonly used is a gene that codes for beta-galactosidase, or beta-gal. And so what happens, the gene is the LAC-Z gene. You might have come across this in regards to the LAC operon at some point, but the LAC-Z gene codes for the enzyme beta-gal. If we then grow the bacteria on medium containing a substrate named X-gal, or known as X-gal, the enzyme can convert the X-gal to a blue pigment. So the point is, if you have an intact plasmid, like we see toward the left, those colonies will produce a blue pigment. And you're not really interested in those, because what you're interested in are plasmids that have had a piece of DNA inserted into them, as we see toward the right. So the green portion, that's a piece of DNA that has been inserted into the plasmid. And when that happens, that will disrupt that LAC-Z gene, because it's inserted right into the middle of the LAC-Z gene. So when that happens, the LAC-Z gene will not be expressed. It will not produce beta-galactosidase, and therefore you end up not producing that blue pigment. So all these colonies shown in white here, those are from bacteria that have had plasmids that have taken up a piece of DNA. So those are the ones you would want to focus on. I don't want to dwell on this, but I just want to point out, if you want to separate, so imagine you've got all these bacteria. They've contained both a main chromosome, shown in white, and plasmids, shown in yellow. If you want to separate those pieces of DNA, you can simply centrifuge with a cesium chloride gradient, and so you end up separating your slightly smaller, lighter plasmid DNA from the main chromosomal DNA within those bacterial cells. Now let's just take a few minutes and discuss creating a cDNA library. So cDNA refers to complementary DNA, and essentially what we're going to do is focus on creating a library that represents, again, just the coding portion of the genome. So just all those parts of DNA that are expressed and used to produce RNA. So what you do is some basic steps. So step number one, you want to isolate the RNA from your tissue or cell type that you're interested in. So you isolate all the RNA, and it turns out that, again, what we're ultimately interested in is mRNA. And mRNA is actually the least abundant of all the types of RNA found within cells. So how can we isolate or separate mRNA from things like tRNA and ribosomal RNA? Well, it turns out that mRNA, recall, will be processed and produce the, among other things, it will produce the poly-A tail. So what we can do is use a substrate that contains a stretch of T's. That's listed toward the bottom as a LIGO-DT. That's just a stretch of deoxythymine. So a substrate that has lots of T's, they will then bind to the A's, the poly-A tail. They will then retain the mRNA, and all the other RNAs will essentially run right through your substrate. So now we've isolated just the mRNA, and what we want to do now is convert that to a corresponding piece of DNA. So step two is we contain complementary DNA from the RNA. How do we do that? Well, first of all, you would add reverse transcriptase. That's going to create one of the strands. Let's backtrack. It will take the mRNA and use that to produce a complementary piece of DNA, but again, that will just be a single strand of DNA. You can then add DNA polymerase, for example, Taq polymerase, we'll talk about that in a second, which will then create the second strand of DNA. So you ultimately end up with double-stranded DNA that, again, just reflects the mRNAs within cells. So these strands of DNA just reflect those parts of the genome that are expressed in the particular tissue or cell type that you're working with. Step 3, then you would then just transform bacteria, much like we discussed previously. So add these pieces of DNA into bacteria. So now you have a bunch of different bacteria, each containing different portions of complementary DNA. We keep talking about plasmids as vectors, and that's fine. They're very commonly used. But I just want to point out that there are other types of vectors. And what you notice here on this list is that often the vector that people will use to work with DNA largely depends on the size of the DNA that they want to work with. And now let's briefly talk about a very commonly used technique called PCR, or polymerase chain reaction. You have likely heard about this before. This is simply a way to create millions, if not billions, of copies of DNA from potentially a very small sample. This was developed in the mid-80s by Kary Mullis, and this has had huge implications for molecular biology and biotechnology in general. So essentially, it's just DNA synthesis in a test tube. And to do that, you need just a few basic components listed toward the bottom in yellow. So first of all, you need a polymerase, a specific DNA polymerase referred to as Taq polymerase. It's called Taq polymerase because it comes from the bacterium Thermus aquaticus. This is a bacterium that can grow and thrive in very hot environments. And that's crucial for this process, as we'll see, because the polymerase has to be able to work at high temperatures. You need all 4 of your bases, your A's, T's, G's, and C's. So all 4 of your deoxynucleotide triphosphates. You need some sample DNA, and it could come from just about anything. It could come from samples from a crime scene. It could come from individuals that want to have genetic testing. Just whatever DNA you're interested in amplifying or copying. And for DNA replication, don't forget you need some primers. So here we're talking about short pieces of DNA. Unlike replication in living cells, which use RNA as a primer, in PCR, the primers are short pieces of DNA. And those are important because those sort of define the region of DNA that will be copied or amplified. The steps in PCR are essentially as follows. You mix all your components together, and then you raise the temperature quite hot, as we see here. So step 1, raising the temperature. What that does is that separates the 2 strands of DNA. Then step 2, you lower your temperature a little bit. That then allows the primers to bind to or to anneal to your sample DNA. Raise the temperature a little bit, step 3. Then your DNA polymerase, or Taq polymerase, can do its thing. It can create pieces of DNA that are complementary to each of the strands that were just formed earlier. And essentially what you do then is you just repeat those steps over and over and over again. And so each time you do that, you double the amount of DNA in your sample. So another way to think about the process is illustrated here. So again, step 1, you take your sample DNA along with your other components that you add together. You heat it up. That separates the 2 strands of DNA. Cool it. Your primers anneal, shown here in yellow, or they bind to the piece of DNA. Warm it up a little bit. DNA polymerase comes along and adds bases that are complementary, shown in red here, to your sample DNA. So after that 1 cycle, you've just doubled your DNA. Repeat those cycles. Every time you do that, you double your DNA. So again, it's a way to copy or amplify pieces of DNA from whatever sample you're working with. And again, the region or sequences that you copy are based on the primers that you use for your PCR reactions. And so the final technique I want to talk about in this chapter is in vitro mutagenesis. There are various ways in which people can introduce mutations into specific positions in DNA. So for example, we can use PCR to change just single bases and create changes in single amino acids and proteins, or we can delete portions of genes, again to create mutations. So for now, we're just going to focus on PCR-Directed Mutagenesis. So again, what we're talking about is creating specific base changes within a gene of interest. So let's say, for example, we want to convert an amino acid, one amino acid to another. So let's say, for example, we want to convert a leucine to a phenylalanine. So toward the left here, we see a plasmid. In this case, we're working with a single-stranded DNA plasmid. And let's say part of a particular gene has the sequence CTT. And our goal is to just change one of those bases there. So what we can do to begin with is treat this plasmid, again it's DNA, with methylase. What that'll do is that will add methyl groups to the DNA. So our plasmid now becomes methylated, and we'll see why that's important in just a few minutes, but that's now shown in yellow. So now we have a methylated plasmid, and our next step then is to basically run PCR on the plasmid. So of course we need all of our ingredients for PCR, including a primer. And the primer is shown toward the bottom here by the white arrow. And our primer again is going to be complementary to a specific region in our gene. But notice what we've done here. We've added a primer that contains a single base that represents a mismatch. So instead of this primer having a G in this spot, like you would expect if that's a C right there, we're using a primer that has an A instead. So that's on purpose because we're using this to introduce a single base change in this particular gene. So again, you add your primer and your other components for PCR, you run PCR, and then toward the right we have now a double-stranded plasmid that consists of our methylated DNA along with normal DNA, shown in white toward the outer edge. So again, the newly synthesized DNA is not methylated, but the original strand is methylated. The next step then is to take this plasmid, use it to transform bacteria, and essentially what will happen is once inside the bacteria, enzymes will digest or break down the methylated DNA. So all of the methylated DNA is digested or broken down, then the bacteria will synthesize a complementary strand to the non-methylated sequence, right, the outer sequence in white here. So ultimately what do we end up with? We end up with a plasmid that now contains a new or a different sequence of bases here. So now we've converted this one base pair right here from a C now to a T. So now we've introduced a mutation, a specific mutation, into this particular gene. And this can be used for a whole host of functions, to see the function of various parts of proteins, or even commercially to produce proteins that, for example, even work better when they're added to laundry detergents, for example. So that concludes our brief discussion of gene isolation and manipulation.